Okay, we're going to go through the marking of the paper that I attempted. Um, I will only highlight the places where I've lost marks, just to kind of save time. Um, Nobody wants to see me just going on about what I got correct. Okay, so let's go through this. Right, so first question, what I've done is I've just indicated with stars which parts of the paper I got wrong and where I lost marks. So let's go there. So the first place I lost marks was question 2 and C. Question 2 and C. Yeah, I got 2 marks out of this. So I lost a mark here. 2C part 1. Chloride ions leave. So I said chloride ions move from epithelial cell cytoplasm. I got the osmosis mark. Okay, so about I can, what I'll do is indicate. No, I want to scribble. Okay. Yep. Right, so I'll just. What is that? What is that? I want to be writing. Okay, so 2C part 1. Chloride ions leave the cell. I think I must have got that one. Chloride, sodium ions. I didn't get about. I forgot about the sodium ions. Increasing the solute concentration in the mucus. So So what I did was, I said ions move from epithelial cell cytoplasm. So I did do that. I did ions move from epithelial cell cytoplasm, but by facilitated diffusion, and then I just went straight to this causes water to move by osmosis. So what I forgot to say was this increases the solute concentration, so I forgot to give the reason why the water is moving. Okay, which is something I will try and do better. Okay, so yep, okay with the mark scheme. Mark scheme goes through step by step, so that's fine. All right, now let's move on. Okay, so in the next one, I liked this question. So what did I miss here? So a triplet code. I think I got triplet code. Um, yes, I know what I did wrong in this question. So I started with triplet is non-overlapping. Every 3 bases equals 1 amino acid, but I didn't give examples. So it does say in here, explain how the information in the table demonstrates. So I got a little bit overconfident. I knew what the question wanted. It wanted triplet. It wanted degenerate, right? So triplet, non-overlapping, and degenerate. But what I didn't do, I didn't give examples from the table for each one. Okay, so every 3 bases equals 1 amino acid. I should have taken the first codon right here, or triplet, ATT, and said that ATT gives us the amino acid I. Okay, so that was an interesting reason to lose marks there. Then in question 3, I forgot to say circular DNA. So here it says circular DNA, or do I need to pick that again? Hopefully not. Right, it says a large, oops, what's going on here? It says large circular DNA, and I rather went for saying loop. So I don't know, I probably wouldn't get that mark. And once I see things on a mark scheme, I should probably start training myself to say those things. Okay, but yeah, so I got 2 marks for other things, but yeah, all right. And then we had the calculation question, which this one was a little bit odd, because it said the steepest part of the graph, and what I assumed was that it was this whole steep part here, but what they wanted, I think, was the bit of this graph that was the actual steepest point, and so it was really between these 2 points, right there. So they wanted the specific gradient for that particular very steepest part of the curve of the graph. Um, but even though I did use like from this point to this point, I did get my answer within the range. But it's just something to watch out for when when they say, what did the question say? The question said um, the fastest rate of uptake. So they really meant the steepest part, and it wasn't just um, that steep line there. It was the very steepest line that we could find. Okay, which I'm fine with. So far we've got into question 3, and I don't have any major problems with the mark schemes. I've lost marks for my own mistakes, and that's fine. Okay, let's keep going. No issues there. Antibiotics was fine. Question 4, photosynthesis. It was 5 marks. I got 4. So one would think I should know photosynthesis quite well. So what did I do wrong here? Um, Question 4A. Right, so it didn't have loads and loads of options, so it was quite a specific mark scheme, just 5 marking points for 5 marks. What did I forget to say? I did say NADP was reduced. I must have said ATP was produced. Electrons to replace lost by chlorophyll. I got that. Photolysis, splitting of water to produce. I think photolysis of water produces oxygen and electrons. I think I didn't say hydrogen ions. So I missed that mark because I did not. I forgot to talk about photolysis producing the hydrogen ions. Okay, but it also allowed a correct equation, so maybe I could have done that. Okay, so I lost that mark. All right, and I think that was okay. Ah, yes, this question that confused me, it did get me because I did get that one wrong. So it said, which of the following is the immediate product of the light independent reactions of photosynthesis? I went with glucose because I felt GP was one of the intermediates, but GP was the immediate product of the light independent reactions of photosynthesis. And glucose is made from that. So that's where I went wrong there. Technicalities, potato, a little bit. Okay, let's keep going. Right, next one. Peat bogs. I like this question, but I did lose a mark on it, so 5A. Pollen preserved in peat bogs. I believe I got that one. Plant species can be identified from its pollen. I think I must have got that one too, towards the end, yeah. Observing the pollen can tell you what species it is. Okay, that's fine. So I think I got that one. Climate affects the type of plants. I think I didn't get that one. The depth of peat correlates with period of time. I believe I got that one. Changes in pollen over time indicate changes in climate. So I think I fell into the trap of just describing how this technique works, looking at analyzing peat bogs. But I think what I missed was this part here, which is how can this technique give us information about climate change. So those 2 marks on the mark scheme, I missed those. Okay. Right, all right, let's keep going. That was fine. And then, yeah, I did this. I found this one a little bit odd. Okay, so we got those forests. I thought I was onto it because I was looking at the respiration values. So correct values for carbon released by boreal forest and deciduous forest. Oh no, that's the calculations. The calculation was fine. But it was the next one, 5B part 2, the ratio of NPP to GPP is higher in deciduous forests. I thought that they wanted a little bit more to do with this respiration thing, but it turned out to be a bit more descriptive. NPP is higher, more of the carbon fixed is used to produce biomass. I did not get that one. And in deciduous forest, more carbon dioxide is removed by photosynthesis than returned by respiration. So I believe I got this third mark, but I didn't get those two. The ratio of NPP to GPP is higher in deciduous forest. The TD ratio is higher. Well, I believe I got that one there. TD ratio is higher. That means GPP to are closer together. Well, it's higher, more the content is higher. Yeah, I think I might have got the more carbon dioxide removed by photosynthesis than returned by respiration. So I don't know. I think I'll stick with the 1 mark there. Not convinced by the other 2. Right, graph was fine. Calculation, Dr. Bhavsar, this is shocking. Did the calculation okay. Forgot the per seconds unit. So remember that you use your unit here, but if you're working out rate based on the time units here, well, that's not per second. That must be per minute. That's per minute. So if we're dividing the y unit by the x unit, we must make sure that the y and the x units are incorporated in our units down here. Terrible. Okay. That was okay. I lost a mark here. Explain how, or a couple of marks. Yeah, I did not like this question. I think I just did not understand what it wanted. But if I'm looking at the question now, explain how the results of this investigation could be used to identify individuals at risk of CVD. So now that I look at that, the mark scheme does make a little bit more sense. We're using the results to say who could be at risk. So how does this table provide information about who's at risk? So yeah, anything outside of the range of without CVD, anything outside of this range, therefore identifies people who are more likely to have CVD based on their fibrinogen content, and therefore what we're looking at is the data differences between the with CVD and the without CVD, basically. So that's where I went wrong with that one. Let's just have a quick look at that mark scheme. Fibrinogen concentration is higher in individuals with CVD. Shall we zoom in a little bit on that one, can I? Okay. Right. Compare an individual's fibrinogen concentration. Yeah, so that seems reasonable, but I don't know if I would say that. That doesn't seem obvious to me. No overlap between fibrinogen concentrations for those with CVD compared with those without. So yeah, so there is a significant difference, which is why I was doing all the calculations. So clearly I must have said something, or I should have said something more general, maybe. So I did all those calculations, but perhaps I didn't use them in my answer. Yeah, so I didn't say that. And that this does seem reasonable, but I don't know how I can change my thought process to be saying stuff like that. Right. Okay, and then let's move on. Next one was okay. Lost a mark. Explain how changes in fibrinogen concentration might be a risk factor for CVD. I said increased concentration of fibrinogen. I said increases conversion to fibrin. Increasing blood clotting. I forgot to say this. Yeah, this is the kind of stuff that I tend to forget to say. So it's just completing that story. All right, so we start with fibrinogen concentration, and it should be point without gaps. That takes us to CVD. And I'm guilty of not completing that story, that third step. So we've got blood clots, but how do blood clots cause cardiovascular disease, right? Sure. Okay. Then we've got the structure of collagen, factual details, fine, just missed one out there, polypeptide chains, 3 chains, small R groups was fine. Let's just have a quick look at that mark scheme. 3 polypeptide chains, repeating amino acid sequence, chains. I did say cross-linking. I did say 3 polypeptide chains, though maybe not. I said triple helix, but perhaps I should have said, well, I did say 3 polypeptide chains. Primary structure described as a repeating amino acid sequence allow, and I did say small R groups glycine. So I think I've been a bit harsh with myself there. I did say 3 things, but I think I was just a bit upset with myself for not saying everything. Right, next. Okay, that one was fine. Yeah, this one was a bit odd. I think I lost 1 mark because here the most obvious things about the strains and their association with the cervical cancer or the genital warts, that part was okay. But then for the 3rd mark, they wanted to say that the genital warts numbers, if you look here, genital warts numbers are higher, right? So the genital warts, we got 67, 50, oops, that's 51 now. Oh no. Genital warts 22, cervical cancer 55, genital warts 67, 22. Yeah, it was just about, wait, let's just go to that mark scheme because now I can't remember what I... Okay, right, so it says, for the strains shown, greater percentage of cases of genital warts are associated with HPV than cervical cancer. So cervical cancer generally has lower numbers, generally speaking. So I did not catch on to that part there. And I didn't make a quantitative, so that's another thing, like what I'm seeing for the comment questions, for all these comment questions, there does, not every single time, but usually there is credit for doing some kind of mathematical comparison. So that's something I'm gonna try and do a bit better in future comment questions. Okay, so let's keep moving. Then I did not do well on this question, though I think I said some things in a different way than what the mark scheme had. So I think I'm not giving myself, so what the mark scheme said was both vaccines will provide immunity against cervical cancer, whereas Gardasil will also provide immunity against genital warts. Now, in my answer, I said Gardasil will prevent both genital warts and cervical cancer, but Cervarix would only prevent cervical cancer. So I think I would get those marks. Yeah, and I did miss the top mark, but I don't know. That just seems a bit obvious to say. But maybe I should work on, making my first point in a question, even if it's obvious, just making my first point in a question something that seems obvious, and then adding value with the other points. Because sometimes the first marking point is very, very obvious, and we avoid saying it because we think it's too much, too on the nose, but maybe it's just worth saying it. How much time does it really take? Okay, moving on. The T cell question was fine for me. Meiosis, the stem cell one was okay. Sex linkage was, sex linkage. Yep, for the sex linkage question, it was on my mind to say it, but then I didn't. And it was the second marking point. So I described the first marking point, or state the first marking point, but I did not add this, that because of these genes located on the sex chromosomes, it tends to cause characteristics or traits that are linked to being male or female. So yeah, or saying how this affects the inheritance. So for 2 marks, I should have seen that coming, and I didn't, but I will remember that now. All right, then we're on to question 9. And I did not, this is the first mark scheme that I think, you know what, they took the piss a little bit. um, because uh, I think all the changes were very subtle, but some changes they're calling not significant or no change, whereas other ones they are calling, or the a little bit. So for this marking point, I said that there was a very slight decrease in the carbon, but they have gone with no change. Okay. Um, Adding cow dung causes a slight or no change to the increase or slight no change to the increase of nitrogen. So here they've gone with slight change. Okay. Um, and here they've said no significant effect, which I don't know. Without error bars or things like that, I'm not sure that we're supposed to be saying that something's significant or not significant based on arbitrarily judging numbers to be small or big. I thought that that was something we are not supposed to do. So, do not like that one. But I can see where they're coming from. I did do the calculations a bit in the wrong way, so I did percentage changes on top of percentage differences. So maybe my life would have been difficult if I just because it was already in percentages, these numbers, because it was already in percentages. I didn't have to work out percentage changes. I just had to subtract, right? So just subtract 41 and 54, and just subtract 35 and 48, and maybe that would have given me numbers that would have helped my analysis be more along the lines of what they were expecting. Okay. Right, and this one was fine. Generally speaking, I think with a question like this, it is very, very off outside of the box. So there's not really a practical where you can memorize your way out of this one. You're just gonna have to think of something that makes sense. And so the mark scheme was accordingly quite general. So I think as long as you referred to things, how are you going to set up your different piles of compost? You're going to take samples at regular intervals. You're going to set up different carbon nitrogen ratios or measure them. Um, but I've given myself 4 out of 6 because I didn't. I don't think I got these last two marking points. So I didn't talk about what I was going to keep the same in terms of other factors that could affect the rate of the succession. Um, and and I didn't talk about repetition of sampling. So just remember back to when it's a question like this, when it is very, very off spec and you have to create a new practical, always go back to your basics. Talk about what you're going to change and how you're going to change it. Talk about what it is that you're going to measure, what's going on there. Talk about your dependent variables, and don't forget to talk about your control variables, i.e., what you're going to keep the same. Okay, and let's simplify these questions for ourselves. All right, then we get this last question about the elephants and whether they're different species or not. We've got lots of information. We have another definition, which was fine. We have a Hardy-Weinberg calculation, which was okay. And then we have this final 6 marker. I think this one was okay. I didn't. I think I did all right in terms of uh, using the information. So they've given us a lot of information, and as long as we use it, I think we're pretty much there. But because it's talking about species, I think there's a few things that were on the suggested mark scheme that potentially I could have added. So, for example, the references to the locations. I didn't talk about the locations. And yeah, I didn't think about how many elephants were used in the DNA analysis, because the more elephants that are used, it does affect the validity of the data. But other than that, I was okay with this question. Mark scheme seems fine. It's pretty straightforward structure wise. And there we have that last question. And then we had speciation question, which was okay. No surprises there. So that's the 2017 paper. As you can see, we have some work to do, or at least I do, in terms of developing my exam technique. But this is 2017 paper. We've got lots of papers ahead of us. So hopefully, by carrying on the process, by learning from the mistakes, we can just make gradual steps forward. And by the time the exams come, we should be much, much more refined in terms of our technique. Okay, and much better in terms of our success results. Okay, thank you.